Hello friends, welcome to SAM Biology class. Today we shall discuss about RNA interference. Before that, I request all of my friends, those who have not subscribed my YouTube channel, please subscribe. RNA interference is a biological process in which RNA molecules are involved in the sequence-specific suppression of gene expression by double-stranded RNA through transcriptional or translational repression. In 2006, Andrew Fire and Craig C. Mello were awarded Nobel Prize in Medicine for their work on interference in the nematode worm Sinorhabditis elegans. The central components of RNA interference pathway are small interfering RNA and microRNA. This pathway involves two mechanisms. One is post-transcriptional, post-transcriptional gene silencing, and the other one is pre-transcriptional gene silencing. In the post-transcriptional gene silencing, mRNA is degraded, as a result of which translation is prevented. Whereas in case of pre-transcriptional silencing, an enzyme complex catalyzes DNA methylation at genomic position complementary to the complex small interfering RNA or microRNA. RNA interference is also known by other names such as post-transcriptional gene silencing, co-suppression, and quelling. RNA interference is an RNA-dependent gene silencing process. It occurs naturally in eukaryotic cells. This process is controlled by RNA-induced silencing complex RISC and is initiated by short double-stranded RNAs in cell cytoplasm, where they interact with the RISC catalytic component known as argonaute. RNA interference can be initiated by exogenous double-stranded RNA, such as those are coming from an infection by a virus with RNA genome or from laboratory manipulation. If this is the case, then the exogenous double-stranded RNA is imported directly into the cytoplasm, where it is cleaved by the enzyme dicer in order to release a short double-stranded RNA known as small interfering RNA. Small interfering RNA are small double-stranded RNAs having 20 to 25 base pairs. The 5' end is phosphorylated and the 3' end has a 2 nucleotide overhang. In addition, RNA interference can also be initiated by endogenous double-stranded RNA such as microRNA. MicroRNA genes are transcribed in the cell's nucleus into long primary transcripts known as primary microRNA, which has one or more characteristic stem and loop structures. The stem and loop structures are recognized and cleaved by a nuclear microprocessor in order to release a precursor microRNA which has about 60 to 80 nucleotides. This nuclear microprocessor is a heterotrimeric complex consisting of one molecule of DROSA, which is an enzyme RNase3, and two molecules of DGCR, which is a double-stranded RNA binding protein. Then the precursor microRNA is exported into the cytoplasm through the nuclear pore complex with the aid of a protein exportin 5. After its export into the cytoplasm, the precursor microRNA is cleaved by the enzyme dicer into short double-stranded RNAs known as microRNAs. The phenomenon of RNA interference thus include endogenously induced silencing effect of microRNA and foreign double-stranded RNA-induced silencing. Mature microRNAs are structurally similar to small interfering RNA. However, before reaching maturity, microRNAs must first undergo extensive post-translational modification. MicroRNAs are obtained from primary microRNA, which are transcribed in the nucleus. Small interfering RNA is derived from exogenous source. So both the exogenous pathway and the endogenous pathway converts at the RNA-induced silencing complex. After formation, small interfering RNA are separated into single strands and are integrated into an active RISC by an RISC loading complex. RISC loading complex RLC. RLC include dicer and R2D2. The active components of RNA-induced silencing complex are endonucleases called as Argonaute proteins. Out of two strands, one strand is known as sense strand, which is degraded during RISC activation, and the other strand, which is known as antisense strand, remain integrated into the RISC and direct gene silencing. After the formation of mature RISC, the antisense strand direct gene silencing. This is achieved by base pairing between the antisense strand and a mRNA molecule. So here you can see that there is a base pairing between the antisense strand and the mRNA. After base pairing, Argonaute proteins, which are the active components of the RNA-induced silencing complex, catalyzes the destructive cleavage of mRNA. So in this way, the mRNA is prevented from being used as a translation template. Differently from small interfering RNA, microRNA, after integration into an active RISC, scan the cytoplasmic RNAs for potential complementarity. In this case, the microRNAs binds to the 3' untranslated region of mRNA, thereby preventing the access to ribosome. So in this way, translation is inhibited. In addition to this, microRNA along with the risk also catalyzes the cleavage of mRNA. So friends, this is all about RNA interference, which is a mechanism that is used by different organisms to carry out sequence-specific suppression of gene expression.